Thank you, Professor Eddie, for the opening speech. Honorable speaker, moderator, ladies and gentlemen, we have now reached to the main agenda today in this guest lecture. Our presentation and discussion session will be led by our distinguished moderator. Please welcome Dr. Sinto Wayuning Ardi. I'm delighted to introduce our compelling moderator. Dr. Sinto got her PhD from Tokyo University. Her research interest has been focusing on the plant tolerance mechanism to abiotic stresses using physiological and molecular approach. Without further ado, to Dr. Sinto, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Mas Ade, for the kind introduction. Professor Edi Santosa, Professor Dewi, Tsugama Sensei, and all honorable guests. This is my great honor here to be moderating the session. I feel a bit emotional like Edi Sensei because finally after 14 years, I can invite Tsugama Sensei come to Bogor. When I was graduating, I told him that please come to Bogor one day. Then after 14 years, it can happen. So I'm inviting Tsugama Sensei to accompany me on the front. Tsugama Sensei, please come here. So I would like to introduce Tsugama Sensei to the audiences. Maybe some of you have joined his lecture in the previous event organized by the Faculty of Agriculture. So, but maybe some of you the first time to see him. So, Associate Professor Daisuke Tsugama, I call him Tsugama Sensei in the more casual way, graduated from Tokyo University in Japan for bachelor, master, and PhD. Soon after his graduation in 2013, he became a visiting scholar in the Pennsylvania State University in the United States. And at the same time, he was also the GSPS Research Fellow in our Graduate School of Agricultural and Life Science in the University of Tokyo. In 2015, he returned to Japan to join Hokkaido University as an assistant professor and a researcher faculty of agriculture. And happily, in 2019, he returned to our lab in the University of Tokyo as an associate professor. The current name of the center is ASEAN Research Center for Bioresource and Environmental Sciences. Tsugama Sensei is a very productive researcher. You can search his publication in the Scopus and Orchids. Currently, his age index is 18. So today, he will share to us about the next generation sequencing. Most of you maybe already know the Illumina or nanopore technology, but how about the data analysis and interpretation? We hope to have more insight from Tsugama Sensei. So Tsugama Sensei, please, the time is yours. You have practically 1 hour to have your lecture, but please feel free, and this is your pointers.
Selamat pagi. Pagi. Terima kasih for having me here like this. I'm really happy to be here finally and meet many people in person. So thank you again. And thank you for the hospitality. I'm very happy with this trip. Everything is taken care of. 新藤さん、レビーさん、エディさん、and many people, and I'm very happy again. Thank you very much. Today, I'm gonna talk about next generation sequencing, data analysis and interpretation. And Eddie-san said, you should understand this one, but I'm afraid I may be using some difficult words for difficult things. So I'm sorry if I don't understand. And please feel free to ask anything about the contents of this talk. and anything related to, for example, a course in our university, everything. I'm also happy to contribute to our collaborative activities and research. So, please feel free to talk with me. Okay. So I will begin the talk of mine. 進まない。あ、OK. OK, responding. So this is the outline of this talk. So in the beginning, I will talk about what is LZS and briefly looking at sequencers and applications. And in the second part, I will talk about NGS data analysis. And in the third part, I will talk about applications to our studies, case studies. And I'm afraid I'm sorry, but I have not practiced well. So the time may not be enough for the third part. And this is to kind of fill the time. Okay. So I will start with the first part. So I saw Shinto-san's material for the sequencers and NGS, and it was better than mine, but this is for reminding you of the sequencers. The next generation sequencing is the sequencing performed by the next generation sequencers, which generate a lot of data with one run. And this is the latest model for the Illumina next gen sequencer, NovaSeq 10. And so this is the maximum volume, maximum data generated by 48-hour run. So this is big, like this. And the characteristics of the Illumina sequencers is that they can generate short read like this to paired end with 150 bases or 100 bases. And this sequencer is capable of generating high fidelity read or very accurate read. The next one is MGI-DNB-seq, and so this is relatively new in next-gen sequencers, and this can generate 6 terabase per 30 hours, and so this can also generate short reads like Illumina's one, and accuracy is a little bit lower than Illumina's one, to my understanding, but this is still accurate. So the next one should be the Oxford Nanopore Technologies sequencers. And I think this is the most popular one, Minion. And the point of this one is the smallness like this. So we plug this one to the laptop computer and we can run this. I have ever used this one personally. And so this can now generate a lot of data than before, more data than before, and after 72 hours. And so the point is this one, long read. So the average length of the read is like 25 kb. So very long. How to prepare DNA is important in this case. We have to protect it from being degraded during the purification process. So DNA isolation method is also important. And Gridion is a sequencer for setting Minion flow cells like this, so up to 5. So we can set 5 Minion flow cells and generate the data at the same time. So the data amount should be like 5 times more than this one. And Promethion is the last one. And for this one, smaller flow cells are used, but this can generate 200 gigabase for 72 hours. And the read length is similar to Minion. So this is more like for high throughput analysis. and a lot of samples can be analyzed at the same time using many flow cells. The next one, the last one should be PacBio sequencer radio. So this is the latest model from PacBio, and so the throughput was improved, and at 90 gigabase can be generated for 24 hours. And this is also for long reads, so 20 KB up to 20 KB per read, and the high fidelity mode is used. So I think this is most suitable for determining the genomic sequence of plants in de novo. And so this is from PacBio website. So PacBio is prioritized in this list, but this is a comparison of the characteristics between these sequencers. And because this is from PacBio, a lot of good things are given for PacBio. But PacBio is most expensive, so the price is the problem. So, but this is most suitable for determining the genome again. So if we have budget, then we can use PacBio. And nanopore sequencing is, I think, very reasonable or can be performed with low cost. So according to our budget and other situations, we can choose appropriate platforms for NGS. I usually use short Illumina sequencer because the cost is becoming lower, and I'm kind of familiar with the analysis methods for Illumina RES. So, so next are applications of NGS, general ones. So RNA-seq is, I think, most kind of popular application of NGS. And RNA-seq is a transcriptome analysis using NGS. And this is a flow, simple. So RNA will be extracted, messenger RNA will be purified, and messenger RNA will be then sheared, degraded, and cDNA will be synthesized, and next-gen sequencing will be performed. In our lab, only this step will be performed, and the other steps will be performed by company. We will send the samples to a company and library. The other steps are for library construction and sequencing, and they are both done in the company. And so each contig derived from a de novo assembly of RNA-seq reads corresponds to each kind of transcript. So if we get data, then we can generate contigs corresponding to messenger RNA sequencing seeds in that species. And this is a benefit of RNA-seq, I think. And expression levels of the transcripts are evaluated with FPKM, RPKM, or TPM. So these are normalized expression values for messenger RNA or transcripts. And I will talk about these a little in later slide. And so they are usually used for further analysis such as clustering and Go or geoterm analysis. This is an example of RNA-seq data use. So this is plant public RNA-seq database, which I introduced in the talk in the last week's workshop. And this is a website showing RNA-seq data from a lot of Arabidopsis samples. And this is my favorite gene, BIP1. I have been analyzing this gene for more than 10 years. And the point is that collecting RNA-seq data, we will be able to show those data using these plots. Nice. I think this is nice. And so this website allow us to show the expression level of the gene of interest like this in mutants over rhabdopsis and under different conditions like salt stress, disease, heated, whatever. And in this website, we can also check the expression level of that gene in the control samples at the same time with these stressed samples or mutants. So this is the benefit of this website. Anyway, so what I'd like to say is that RNA-seq data can be useful like this. So we can manually do this kind of analysis if we get RNA-seq data. So it is useful. And another example is GPS, genotyping by sequencing. And this is, as it says, genotyping by sequencing, by next-gen sequencing. The first example given here is RAD-seq, or restriction site-associated DNA sequencing. I will introduce this later. And GRASS-DI is genotyping by random amplicon sequencing direct. I will not explain this one, but this is based on the random amplification of genomic region by PCR, followed by next-gen sequencing. So amplicon will be sequenced. This one is MIG-seq and multiplex inter-SSR genotyping by sequencing. And this is also based on PCR. And for that PCR, the primers will be designed to attach to the SSR regions. So primers will be within the SSR regions. And using those primers, the amplicons will be obtained, and then it will be sequenced by next-gen sequencers. The last one is whole genome sequencing, ARC-BRES, and this is the simplest one, but very powerful. The whole genome will be randomly sequenced in a shotgun manner. And the obtained data will be utilized for coding SNPs, genotyping directly. And I usually try to collect WGS data for genotyping new cultivars or some known cultivars. This is the example of GBS, RAD-seq, and this is the image. So genomic DNA will be obtained like this first, and restriction digested like this enzyme. To the end of the DNA revealed by the restriction digestion, the adapters will be attached like this for next-gen sequencing. And the DNA will be shared, and the second adapters will be attached to the other end of the to DNA. And sequencing will, with Illumina's sequencer, using the first adapter, will be performed. Here is benefits, and I'm sorry for small letters. But the point is that SNPs or single nucleotide polymorphisms can be detected on a genome-wide scale with a relatively low cost. So only the regions in the vicinity of the ends will be sequenced in RAD-seq, but we can get kind of sufficient coverages for those regions. And those regions will cover the whole genome regions, like this percentage of the genome. So this is good to analyze the genotypes of many cultivars at the same time with low cost. And we use this one, we use the data from this analysis for our PARMILET research also. I'm not introducing the PARMILET study today, but ask me or somebody in IPB if you are interested. Material. So this is another the example of application of GBS for genetic study. And this is GWAS, Genome-Wide Association Study. And this is an image for GWAS. So in GWAS, we assess the phenotypes of many cultivars like this. So we can see some plants have purple colors, dark colors like this, so compared to the other plants. And we also genotype these plants. And by using those analyses, we can compare the phenotypes with the genotypes. And then we should be able to detect the genomic regions associated with that phenotype like this. So this is called Manhattan plots. And the x-axis shows the positions on the genome of this species. And the y-axis shows the effects of that genomic region on the phenotype. So in this example, so this region in chromosome, わからない。1, 2, 3. And this region in the chromosome 4 are associated with the color phenotype. はい。多分、sorry。これは、多分。Sorry, I think I pushed this one so many times. で、this is another application of GBS for genetic analysis. This is called MuteMap. And this is one kind of analysis of bulk to segregant analysis. This is for the analysis like this one. Another example will be introduced in the following slide. But first one is MuteMap. In this analysis, the wild type and mutant like this, assume this is mutant with dark color. And the wild type and mutant will be obtained like this. And they will be crossed to get F1 plants. And F1 will be self-crossed like this, and F2 generation will be generated. And in F2 generation, the phenotype will be segregating like this. Okay. And these wild type plants, plants with the wild type phenotype and mutant plants will be collected separately in bulk like this. And as a mixed manner, they will be sequenced. Not mixed manner, but the wild type bulk will be prepared in bulk DNA sample. And mutant bulk plants will be used for preparation for the DNA samples. And those DNA samples will be sequenced. And the SNPs enriched in the mutant bulk will be detected like this. So this is called MuteMap. Here is another example of PSA or bulk segregant analysis, QTL-seq. In this example, the phenotype is quantitative one rather than qualitative one. So in this example, the cultivars with very different sizes are used, and the F1 plants will be obtained by crosses between them. And F2 plants, in F2 generation, the sizes will be segregating like this. And they will be split into the small plant group and large plant group like this. And they will be sequenced in bulk as before. And the SNPs enriched in one of these groups will be detected like this. So this is called QTL-seq. I don't think the name is important, but this is kind of way of analysis of bulk segregant. So these are other applications of NGS, exome sequencing. This targets genomic regions corresponding to exons to see the SNPs in important genes in more deeply than randomly sequencing the transcriptome or not transcriptome, but whole genome. And AMPLICON-seq targets PCR products to find rare SNPs in genetic disease-causing genes or to analyze microbiota. So very specific regions will be targeted for AMPLICON-seq. And instead, the coverage or depth for the sequencing for that gene will be larger. So we can see more mutations or SNPs for that gene with this method. And whole genome bisulfite sequencing, BSseq, is used with genomic DNA treated with bisulfite ion, which converts an unmethylated cytosine to ratio. So, methylated cytosine will be blocked from being conversion of this conversion. So comparing the DNA samples treated with this ion with non-treated samples, we should be able to map the cytosine, unmethylated cytosine. And we can also get the methylated cytosine sites. And we can see then which site is more easily methylated on the genome. Other examples are available. I'm not giving them. But the point is that how target DNA is prepared. So according to the DNA preparation method, we will name the method and then interpret the data to get the new insights. So from here, the NGS data analysis will be introduced. So this is a simple flow for NGS data analysis. So we first run NGS to get reads. And if reference sequence is not available, then we need to assemble those reads into contigs. And then we map those reads to reference sequences, which can be obtained by this step or from public databases. And yeah, here, genome transcripts obtained contigs, etc. And then we will count the reads for each gene or feature in the reference. And for RNA-seq, differentially expressed genes or DEGs will be detected. And the variant call can be performed, like GBS. And further analysis can be performed. This is how the RNA next-gen sequencing data look like. And this is called FASTQ format, FASTQ. Do you know FASTA format? In FASTQ format, this is the ID of read. So the read ID will start with @mark. @mark is kind of mark for as the beginning of the read data, one read data. And ID follows that. And this is a sequence. And here is plus. Line break here, and plus. And then this sequence follows that. And this is corresponding to this sequence. This is called the quality score. So, the number of the letters for this sequence and the number of the letters for this sequence or the quality score are the same. So one nucleotide has one quality score. And if the quality is very low, we can exclude those reads from the analysis. And this is called multi-FASTQ, I think. This can be called multi-FASTQ. And next, read data follows like this. So we can see a dot mark, and this means that the new sequence data will start from here. Okay. In faster format, we will use arrowhead, right? Arrowhead, would you call it arrowhead? Fast queue, so we will use at mark, and plus and sequences like this. Using those sequences and quality score, we can filter the reads, and so then sometimes we do assembly of these reads. And this is easy image, and more probably sophisticated or complicated analysis will be performed by the program for assembly. But this is for simple understanding. So there should be some overlaps between reads like this, and using these overlaps with certain threshold or criteria, we will join those reads into one sequence. And this is called contig. And assembly is the step to generate these kinds of contigs from each read. And this will be used for further analysis, assembly. For further assembly, we sometimes use, for example, high C sequencing, which artificially attach the DNA fragments next to or in the vicinity of each other together. So if the DNA is sequenced together in the high C sequencing, we can assume that they will be on the same chromosome. We can see the contiguity between those reads. And we can also use the genetic map or the linkage between different regions on the genome for further assembly of the reads or contigs. And if we get reference sequences from databases or the de novo assembly, we will do mapping of reads to those sequences. And this is the image for mapping. And here, the read is shown by this color, and the reference is shown as black, like this. Please assume these are the reads associated with these reference sequences. These can be reference to known genome, known transcripts, and contigs obtained by de novo assembly. I set the numbers of the reads associated to aligned to these sequences by 22 for all of them. So anyway, the read counts for each region of the reference sequence are often used to interpret the data. This is for RNA-seq data analysis, and this is FPKM and RPKM, which are used for as the normalized expression value for gene or transcript in RNA-seq. And here is a calculation method for FPKM or RPKM. So FPKM is equal to RPKM when with or single n. So I mean, these are similar, and the calculation method is the same, essentially. I will not elaborate on how to calculate, but this is based on this calculation method. This is another way of normalizing the read count data for RNA-seq, and this is TPM, transcripts per million, and it's based on this calculation method. And TPM is similar to the copy number of messenger RNA of interest divided by the total copy number of messenger RNA in that tissue or sample for RNA sequencing. And TPM is said to be better than FPKM or RPKM. So I think TPM should be used whenever possible, feasible in RNA-seq in the future. And this is the example of FPKM and TPM. And in this example, I set the numbers of the reads for each gene, or 22. They look different. Like their expression level looks like different, right? Between ABC, contiguous ABC in the left example. And that's because of the difference in the lengths of those contigs, right? The contig B is the smallest contig with the length of this one. And the contig C is the longest one with the length of this one. So, we can see that this is likely to be the most, to have the most strong expression among these contigs. And this looks like the weakest expressed one. And that kind of feeling can be consistent with the FPKM or TPM, normalized expression values. This is another example. So assume that these are different samples of interest. We want to compare the gene expression levels between these samples. And these are the, I think, real FPKM values calculated from this data. And we can see that FPKM and TPM are similar between each other. But TPM is recommended now. I don't remember, I'm sorry, the specific reason that TPM is preferred, but anyway, TPM is said to be more accurate or reflects the situation in the samples. And this is often done to get a fold change of genes between the samples, between different samples. So in this example, sample 2, the gene expression in sample 2 was divided by the the expression of the same gene in sample 1. So this is the relative expression level. And so in this way, we can see that the gene B or contig B is more weakly expressed in sample 2 than in sample 1. Comparing these two. And this is so in this way, we can kind of directly or more clearly show the difference of the gene expression between two different samples. I wanna say though that for detecting differentially expressed gene, some different way is used. We can use TPM for comparing the gene expression between different samples, and we can also compare the expression level, for example, between homologs of each other in the same species using TPM. But for detecting differentially expressed genes between different samples, read count data can be directly used, and some specific statistical distribution can be utilized by specific software. I was thinking about giving the specific names of those programs, but I forgot it. And they are HR or D6-2. These two are very often used to detect those DEX or differentially expressed genes in RNA-seq. Previously I was just using TPM, and one reviewer attacked it. And I was told to use those programs, and now I shifted to use them. So anyway, TPM is useful, but to analyze the DEX or differentially expressed genes, some specific programs are used. I think it's better to use them from the beginning. And this is about variant calling. So DNA variants can be detected or called after reads have been mapped to reference RNA or genome reference sequences. And for genome sequencing, new data are often presented as VCF format, variant call format, rather than whole genome sequence. So this means that if we sequence the whole genome of certain cultivar, we should be able to present the whole genome of that cultivar with like whole genome size. For example, so Arabidopsis has 550 megabase genome. And if we, for example, read Arabidopsis cultivar called Kyoto, then probably we can generate 150 base genome of Kyoto and present it. But usually it is not done. So instead, the variants detected from that sequencing will be given in this format very often. So, I mean, it is sometimes necessary to be not to be scared by this kind of data, looking at this data, looking into this kind of format. And this is a VCF, and I'm sorry, very small, but here is a position of the variants on the genome, chromosome number, position on that chromosome, and ID for that variants, quality, and then genotype for that variant in each of the cultivar like this. Here are 3 cultivars given, and some should have heterozygous allele for this position, and some has homozygous allele for that position, and anything like that. That kind of information is given in this file format. So, when we say, so this genotype was genotyped, this cultivar was genotyped by next-gen sequencing, that usually means that we obtain the VCF file from the NGS data from that cultivar. Okay. And the program should be like Strelka, VCF tools, or something like that. This is the image of the variants, DNA variants called by that kind of analysis. And in this example, variants are shown in like this. And these are these references, and we can visualize the variants code like this on some software like integrated genome browser or something like that. And we can see that in all of these examples, the variants are homozygous for this sample, right? Because all the reads have this kind of variants. For heterozygous, clear heterozygous pattern, probably half of the reads will be showing the variation, and half of the reads are not showing the variation. In that case, we will assume that that variant will be heterozygous for that sample. I think this is difficult, but how long can I talk? Or 30 minutes more? Okay. So I will keep on. I think I can talk all of them, talk about all of them. So in the third part, I will introduce our study using the next-gen sequencing as a real study. The first example, I have 2 examples to introduce. And the first one is about tartary buckwheat. Do you know tartary buckwheat? Do you consume it? No? Okay. Not at all. Sorry for that. Okay. In Japan, not so often. We also don't eat this one, this one so much, but yes, soba. But this is different from normal soba. This is called niga soba, bitter soba. So this was previously used for tea mainly, but now non-bitter cultivar was developed, and the developers are trying to advertise the new cultivar for soba also, noodle. And this is annual self-propagating plant species and not regular buckwheat. This has a high level of rooting, which is good for human health, and low nutrient tolerant and cold tolerant. And this was originated in mountainous places. So that's why this has cold tolerance. They were irrelevant information. And so we were working for improving the ease of deharing in Tartary buckwheat. This is not this is not showing the deharing easiness, but the point is that we had Japanese cultivars where deharing is difficult. So the hull was difficult to remove for this cultivar, and we got a genetic resource with in which deharing was easy. So for this species, we just had to use finger for removing the hulls. So deharing was that easy for the right cultivar here. And so deharing easiness is important because the Tartary buckwheat is used as a powder or tea, as a processed food. So during the processing, the contamination of the hull can be a problem. And cultivar with the easy deharing is more useful for that kind of processing. So we wanted to introduce the deharing easiness into the Japanese cultivar, like this. And this is non-bitter cultivar developed by those people. So the goal was like using this one with more easy deharing way. Non-bitter one with easy deharing. The focus was on the deharing thing, so for this study. And this is the non-rice type or the difficult deharing type cultivar. And this is the rice type cultivar. This is seeing the the region around the hull or the young seeds or pistil or carpel. And this is a section. And the point was that we could detect the cell layer that undergoes the periclinal cell division like this. So because of this cell division, this layer is formed. This is the these are the dividing cells around this region. And these kinds of cells were not observed in this rice type or the easy deharing cultivar. So this was important. This was formed in a specific timing of the floral bud development. So we, according to that observation, we determined on which timing we should sample the tissues for RNA sequencing, and then we did it, and then sent the samples for RNA sequencing. And I got the data for read count and TPM and gave those data to a student in Hokkaido University. This study was done when I was in Hokkaido, and Hokkaido is the northern island of Japan, cold place. And anyway, I gave the data to a student, and I was not expecting that much, but that student was good to find a very important or interesting gene. So she was looking at the gene involved in the development or differentiation of the cells and focusing on those genes. And she found one the missing part in that gene like this. This is showing the RNA-seq read coverage on that gene. And that gene was agamous-like gene. So the transcription factor gene possibly involved in differentiation of the cells. And she returned to me this kind of data. And I was very surprised and got interested in that gene. And we decided to proceed with that gene to further characterize that gene. As I said before, this region is missing in this cultivar, the easy deharing cultivar. So, and that region was 42 bases corresponding to 14 amino acids. And the bottom two sequences are those sequences. And we can see here that I'm sorry, this is small, but this is showing that the easy deharing cultivar lacks the 14 amino acids here, like this, because of the 42 base deletion in the messenger RNA of this gene. And we sequenced, we cloned this gene. The genomic sequence of this gene was cloned, and we didn't detect the 42 base deletion in the genomic region, and instead we detected SNP in the exon intron junction here, acceptor site of the intron, like this. And because of this mutation, and this exon, difficult to see, but this exon should be 42 base, and exactly the number of the expected the number of the base was exactly 42 for this exon. So because of we strongly think that because of this SNP, this was splicing was like this, jumped, and this exon was skipped. As a result, 42 base was deleted in the messenger RNA, and that will truncate the protein like this. We developed CAPS marker using that single nucleotide substitution. And using that CAPS marker, we could distinguish the allele on that region like this. If the allele is AA homozygous, then the PCR product cannot be cut by restriction enzyme. And if that is homozygous, then it is almost completely cut like this. And if that is heterozygous like this, half of it is cut and half of it is not cut. So the result becomes like that, like this. And we genotyped in total 58 plants with known phenotypes. And we could see that the genotype of them is completely linked to the phenotype, like this. I'm afraid the number of the plants genotypes is not very large, but at least for these plants, the genotype and phenotypes are completely linked to each other. So we still think that this gene is a candidate for the gene directly regulating the deharing easiness in totally buckwheat. Another example is garden asparagus male specific gene. Do you know garden asparagus? Vegetable. Okay. Okay. So asparagus, the garden asparagus is Asparagus officinalis, and this is a perennial crop, and this is generally dioecious. Did you know this? Yeah, so this plant has kind of sex like human, dioecious. dioecious means, so this is the example of dioecious, yeah, so individually determined. So this is female plant of asparagus, and this is male plant of asparagus, and we cannot distinguish them if looking at this kind of appearance. But if we look at flowers, we can distinguish them. So on female plants, only female flowers are developed, and male plants will develop only the male flowers like this. So looking at usually looking at flower shapes, we can distinguish the sex of the individual plants of asparagus. And in production, male plants are preferred in general because male plants don't develop seeds which can generate offsprings or younger plants which compete for nutrients with older plants. I mean, this is perennial crop, and the products are like 5 years old or older plants. So, so these young plants will disturb the management of the field, and so this is not preferred. The female plants are not preferred for that reason. And this is unisex flowers of asparagus. And this information was kind of important for the further analysis. And so the unisex flower shapes are different between species to species. So this is specific to Asparagus officinalis. And in asparagus, stamens will degenerate in female flowers like this. And the stamens grow normally on male flowers like this. And pistils grow normally on female flowers, and pistils stops growing stays small in male flowers. And by previous genetic analysis, genotypes of them were known like this. So the female plants or flowers have a small m genotype, and male plants have a big M, small m genotype like this. And this is not naturally occurring, but large M genotype is also available by chance. And chromosomes are identical in morphology. So this is comparable to human way of determining sex. And so this is different from the human chromosome shape. We can distinguish sex chromosome in human, but in asparagus, it's not possible. So according to this information, big M locus contains a small number of genes that inhibit pistil growth and help stamen growth, and that does not affect the whole shape of the chromosome. This is kind of supportive data and about big M plants, and this is called super male, which is not naturally occur. And this is good because offspring of that will be all male. So this is variable in production of this kind of, this is called all male seeds or all male plants generation, population. It is difficult to get this one though. And we just have to wait for natural mutation or pollen culture or something like that. Anyway, we try to reach the M locus gene in asparagus and looked at the genes involved in regulating the pollen development in Arabidopsis. So I was working for Arabidopsis for more than 10 years at that point, and I liked to use Arabidopsis information. And I looked at genes involved in pollen development. And because of the previous studies, these are those genes. And because of the previous RNA-seq data, the genes, homologs of those genes were identified in asparagus. And according to that analysis, these genes were not present in garden asparagus. Looking at the expression levels, we noticed that this gene is expressed only in male plants, male flowers in asparagus. Assume that these are asparagus genes, counterparts of the genes regulating the Arabidopsis pollen development. And the point was that we detected male specific expression for MIB35 homolog in asparagus. We also saw that some of these genes are strongly expressed in males, but these were expressed even in the female plants. So MIB35 expression is more specific to male, more specific or 0 in female. And this is it. I was using FPKM at that point, and this is FPKM data. But we can see that for asparagus MIB35, no expression at all was detected for the floral buds at these stages. Sorry, at these stages. And the female plants, not at all in female plants. This is the earliest stage of floral buds development in male plants, and we basically the expression was detected only at this stage for at least this line. And even if we include spear tip sample, we could see some expression for some spear tips in these cultivars, but not at all in female samples. We cannot see any bar here for female samples, 0. In contrast, AOMS is a downstream of MIB35. And we can see that AMS expression. Sorry. We can see that AMS expression is also very difficult to see. But in AOMS, some expression is here. It's difficult to see, but here is some expression. Yeah, you are close to. So sorry for that. But here is some expression of AMS anyways. But in MIB35, no, 0. We confirmed this result by RT-PCR, reverse transcription PCR. And again, the expression is very specific in AOMIB35 and not that much in AOMS. So we can see the expression in female samples here. This is in situ hybridization, and we could see the expression of AOMIB35 in the tapetum cells, which surrounds the pollen grains like this to help the pollen grains develop further, providing some nutrients and necessary things like signaling molecules. This is the punchline slide. So we did the genomic PCR. This is genomic PCR, so not the RT-PCR or expression analysis. So this is kind of genotyping. And surprisingly, we found that the female plants lack this gene. So we could not detect signals, PCR signals in female plants for AOMIB35. This is kind of control. In AOMS, we could detect the signals like this, even in female samples, but it was not possible in AOMIB35. Very surprising, and I was happy to get this data. And we did the same detection of AOMIB35 using more cultivars, and all of them showed negative results in female plants, like the figure before. And one reviewer recommended me to do the Southern blotting, and I was very reluctant to do that, but using 20 microgram DNA or something like that, I finally did it and got a single bands like this. So this, yeah, this is difficult to see probably, but so these are single bands only detected in male samples. So I was happy to get this data also. And this means that the number of this gene, copy number of this gene will be one, just one in asparagus. And that gene is present only in the female plants. Sorry. Male plants. It's confusing, sorry. Yeah. So this gene is present only in male plants as a single copy. So, we did some further analysis for AOMIP35, but this is a punchline. And this is the very end slide. So this is summary. So de novo assembly, mapping, read counting, and variant coding consist of most NGS data analysis. So don't be scared. These are almost all for the analysis. And how DNA samples or libraries for NGS is prepared is important for data interpretation. So according to the DNA preparation method, everything will be discussed after these steps. So don't be scared again. And this is all from me. And thank you for the attention. Terima kasih.
Thank you very much, Tsugama Sensei. You may have a seat together with me again. And I would like to personally thank you to deliver some simple terminology explanation in the beginning, like contact rates with simple illustration for audiences who are not familiar with this technology. And continuing with the example of how this technology will help to identify important traits. So I would like to welcome any question or comments from the audiences who present on site first. You can raise your hand. One question first, second question, three. So we will have three questions in the beginning. Ibu, please mention your name and your question.
Thank you. Thank you for the nice presentations. Okay, as you mentioned before in the summary that in the NGS, the end up samples is very important to be in data interpretation. So do you, regarding to the metagenomic sequencing, do you have any experience, how much DNA sample at least to get the best result for the NGS? In nanogram or picogram, and can you share your experience?
I think I missed some of your questions. So data amount for genome sequencing for what?
For metagenomics.
Metagenomics. I have actually no experience of metagenomics, but metagenomics should be using amplicon-seq or some targeted region for sequencing. To my understanding, for metagenomics or microbiota analysis or something like that, some targeted regions will be used. And in that case, I think the DNA amount necessary for the analysis will be kind of small, because the target region is small. So even if the next-gen sequencing data amount is small, we can get sufficient coverage for that region. And to my understanding, not that much as like plant whole genome sequencing.
In your experience, what have you done in asparagus, and how many how much DNA you get?
For that analysis,
for that analysis,
that was RNA-seq analysis. And the data were generated by different research group. So I downloaded them and utilized the data. And I don't remember how much data were those. But it was a lot. I mean, the data amount was a lot. So I very thank that group for generating a lot of data, and quality was also good. And the sample quality was also good, I would say. It's not related to your question, but the samples they used, I think, were good. The tissues sampling timing, they used different developmental stages, 3 different developmental stages for floral buds, corresponding to the pre-meiotic stage, meiotic stage, and post-meiotic stage. So they very strategically collected those samples and got a lot of data. So the data quality was good. That was what I felt when I was handling those data. I usually use like 3 gigabase or so for every RNA-seq analysis. And I think sometimes it's not enough for some species like wheat or with species for the species with larger genome. But we just start with like 3 gigabase, and I'd say we can still see some differences in gene expression using that kind of data.
Thank you, Tsugama Sensei. But I also received some questions similar to what Ibu just said. It's more related about the sample preparation in the beginning. When you isolate the genomic DNA or you isolate the mRNA to be converted to cDNA for the NGS, do you know the general requirement of the sample isolation?
Quality.
Quality or amount of the DNA?
So amount is also important, but I don't think it's very much. Quality is very important, and the company always check the quality of for RNA-seq, RNA quality, for DNA-seq, DNA quality, and they always check and say something about it. We usually use kit like DNeasy or column-based kit, and it's sometimes okay and sometimes not okay. We try repeating the experiments.
Yeah, I would say also it will maybe just an additional information for like nanopore technology. You would like to have more proportion of high molecular DNA because the nanopore will have longer reads than the Illumina, right? Yeah. So maybe that's one clue.
For nanopore, I heard that CTAP, traditional CTAP-based method is much better than the column-based kit. The column-based kit will share the DNA, for the column binding thing, and that's not good for nanopore or PacBio sequencing.
Yeah, so basically it depends on which NGS we use. The Illumina will allow shared DNA like produced by kit because it will read short reads, but the nanopore require more high molecular DNA. So CTAP works better in that way. Okay. So I would like to move to the next question or comments from Dr. Willie or Arslan first? Or Arslan first. Thank you, Pak Willie.
Thank you, Professor, for your wonderful presentation. And thank you, Pavili, for giving me chance. So I have round about 4 questions. So, Ibu, I'm allowed? I have 4 questions. So, Sensei, like I started my research from whole genome sequencing and narrowing it down to fragment level right now. I'm looking for SNPs in some 5 genes of local Indonesian fruit called salaka, salak snake fruit. So my targeted genes are ITS, MATK, TRNLF, like these 5. So I'm looking for SNPs. But after my Sanger results, I got some, we called them indels or SNPs like WYR. So it can be C over G, it can be A over T, So like there is a confusion, like I align all chloroplast genome of all of my varieties, like species. That was good, like the SNPs was clear. But when it's come to the fragment level, like gene level, like that was is W, Y, T, something like this happen. So I want to ask you how to analyze these W, Y, S, or T kinds of variations. And the other one, if I'm talking about mapping, so we just map to one reference, or I should use all of that 7 species reference for multiple sequence alignment. So my second question, so if we I was able to assemble from whole genome sequencing to chloroplast of all of my species from NCPI, like secondary data. One of the species was 132 kb, and the rest of was like 157. Like they was full chloroplast, and one was partial. But my senior told me like we can map it to the reference, and it will become the full one, and you can recover the full genes. So what you suggest, like it is advisable to map to read that partial genome, and we can count it as a full genome.
Can you remember the question?
So the first question is like how to handle W, Y in K in in the variant code format or DNA sequence. Ah, you know what those single letters mean as nucleotides, like, and I think it's like isn't it possible to interpret those data as such? Like, W, I don't remember actually.
W means A or T, right?
Yeah.
And the Y means C or T. So
yeah, so I think that sequence can be interpreted like that. Probably it's difficult to determine why that region should be heterozygous, and in that case, so we will get two kinds of sequences for the same gene, and to express those two sequences, and that kind of two letter, I mean, the one letter for two letters can be used, I think. Another way would be separating those sequences using, but it's difficult to determine how to separate them. If we have phase data or pitch sequence, I mean, it's difficult to separate.
the chromatogram, the peak.
Yeah, chromatogram can help. We call phase data if we can distinguish the chromosome or the chromosome, chromosome of the parents of that cultivar or the plants. I mean, for example, if there are two sides of the SNP, and if they are written in the single sequence, then it should be difficult to how to separate the sequences. But if some additional data showing which which allele belongs to which parent, then we should be able to separate them. Unless we have that kind of information, I don't think it's possible to separate those sequences into two or more. Does this answer your question?
So the possibility of this IOPAC code appearance is the heterozygosity of that locus, right?
I would assume so. Yeah. If BCF file is available, it's possible to directly look into it and what the genotype is written in that file.
So you still remember the second question?
What was it? We have a partial genome, and we map it
Okay.
Yeah, now we remember.
I think if I were you, I would try both of those references, like the partial one and longer one. And I would do the mapping twice. One is for the partial one, and the other is for the longer sequence, and compare the results. Like mapping rates can be compared between those two mapping procedures. And I would probably use the one with the higher mapping rates. Or I will just look at both and try to discuss the data accordingly.
So did you have 3 questions previously? Only 2? Yeah, please keep the remaining for now. Okay. So I would like to welcome Dr. Willy for your comments or question. Please, Pak Willy.
Thank you very much, Bu Sinto, for the opportunity, and Tsugama Sensei. Thank you for a very interesting talk. I have 2 questions. The first one, in your example in Tartary buckwheat, yes, you mentioned in that research you develop a CAPS marker, yeah, which is linked to the genes, and then controlling which controls the easy dehaling of the easy dehaling phenotype, right? And then my question is, have you ever used that CAPS marker for screening in a heterogeneous population like F2 or something? And what is the, if you have ever used that, what is the efficiency of that markers?
I'm afraid not. We have never used that marker for real selection.
Okay.
We just used for our.
Okay.
Yeah.
Thank you. And then my second question is for the officinalis, isn't it? The second example. And then can we differentiate the phenotype of the homozygous M, homozygous big M, big M, and big M small m? Is there any difference in between super male and male? Yeah.
To my
Thank
eyes,
you.
no.
No.
No. So unless we cross them and see the offsprings of them, we
Okay.
cannot see the genotype of the parent super male.
Okay.
So we cross them and see. Or if all the offsprings are male, then it's super male. The parent is super male. That's the only way to distinguish the super male plants from the regular male plants.
Okay. Okay. Thank you very much.
Thank you very much, Pak Willy. We can open. Okay, Nining Sensei. Hisashiburi desu, Nining Sensei.
Hi.
Please.
Good to see you again.
To see you.
Yeah. I just want a very basic questions in the bulk segregation analysis. You use phenotyping that is discrete. How reliable is it if you have a phenotype that is continuous? For example, you measure the length of the root, for example. How reliable is it to be if you use that for bulk segregation analysis?
I think so. In the example I was giving, it was like that. I mean, the plant size was used as the example, and it was continuous phenotype rather than the separate one, discrete one. So that example was showing that the QTL sec is for that kind of analysis. I think it's better to show the distribution of the data though. So if the data distribution is like consistent with the Mendelian pattern like this, then we can also think about we just use bulk segregant, but probably I don't know. I don't know, but it can help to separate the groups. I mean, I think the preparation of the bulk is important in those analysis, and 難しいな、でも。 Yeah, for continuous phenotype, I was showing that very extreme, the plants with extreme phenotypes are used, and probably that's the usual way for that kind of QTL-seq or that kind of bulk to segregant analysis. I don't have a good idea about that.
Like how extreme? So
Yes.
only the very extreme, extreme one.
What I think is that so seeing the pattern of the separation should be important in the F2 generation.
Actually, I have a similar question with Nining-san because we develop F2 generation and we, for example, have this normalized data. So you want to choose the extreme left and extreme right for the bulk. And somehow we have question how to separate this data. What is the threshold? Did you decide, for example, 20% left, 20% right, or is there any particular clue to do that in practical?
I can ask the developer of that method. Another way is using histogram.
Yes.
And if the histogram is kind of like this, then probably threshold should be.
Yeah, we're actually doing that and always challenge with how to separate the phenotype to choose the bulk.
Yeah, it's difficult.
Thank you. So thank you, Buning. Any questions from the audiences? Okay, Ali, and another question from Hisham. Okay, Ali first, please.
Thank you, Ibu, and thank you, Prof. Tsugama. All the way on your presentation, you have been using RNA for your research. My question is it working better with NGS more than DNA, or it will depend on what you intend to achieve? Thank you.
You explained about RNA sequencing and also genomic whole genome sequencing. So he was asking which one is better, or it depends on the purpose of the study. Am I right, Ellie? Yes.
Genomic sequencing.
Yeah, whole genome sequencing, you use DNA, and RNA sequencing, you use RNA. So which one is better, or it depends on your purpose?
It depends on the purpose, but if we do RNA sequencing, we can see which transcript is present in the target samples. So probably we can exclude the genes that is not expressed in that sample from the further analysis if we are interested in very specific gene regulating some specific phenotype. And using the RNA-seq, we can also call the variants in the transcript. So we can get the information about the gene expression and sequence variant at the same time using the RNA-seq. So having said that, I think DNA sequencing is also important sometimes. For example, if I don't have so much budget, probably I will try RNA-seq, prioritize RNA-seq over the genomic sequencing. But it largely depends on the purpose. Does this answer your question? Okay.
Yeah, indeed. If you would like to see the differential gene expression, then the RNA sequencing is more preferable. But if you would like to make, for example, reference genome, then whole genome sequencing is more preferable. So Hisham, shall I welcome your question, please?
Okay, thank you for the chance. My name is Hisham. I'm from undergraduate student from agronomy and horticulture. Thank you for the great presentation, Tsugama Sensei. I want to ask 2 questions related about your project in asparagus, male-specific marker. The first question is, can we test the marker of the asparagus for other disease taxa? Have you tried it, and how the result? The second question, the second question is, how many genes that actually control the sex of the asparagus, and how we can choose it, which genes that can represent or that we can detect as the marker of the sex of the plants. Thank you.
Fortunately for your question and unfortunately for me, there were publications regarding your questions. And for the applicability of the AOMIB35 to other taxa or other species, right? In asparagus, some other asparagus species have sex actually, diacy. And AOMIB35 was working for those species as well. So at least AOMIB35 should be conserved between those species, and it can be used as a marker for the male plants for all of those species.
Different species, but still in asparagus genus.
Yeah.
Okay.
If I don't think it's true for different genus. I think the mechanisms of the sex differentiation are very diverse in plants. It can be easily obtained, I think, in natural conditions. So different species have different mechanisms for determining the sex. And for the other questions, one research group which got the high quality RNA-seq data originally say that two genes are involved in determining the sex of Asparagus officinalis. And one is AOMIB35, and the other is SOF, SOFF gene. It's a suppressor of female function. So basically, the function of the sex determining gene in asparagus are two. One is to help the stamen development. If that gene is absent, then the stamen development is aborted. That's AOMIB35, probably. And the other function is to suppress the female pistil growth in male plants. And that function is dependent on SOFF gene, they think. So in the current model, there are two genes in that BigM region. I'm not sure whether two are enough, but at least a small number of genes are involved, and the associated functions are two. Stamen development promotion and suppression of the pistil growth. And I think the previous histological analysis and genetic analysis are also important. We know that male phenotype is dominant over the female phenotype in the asparagus. And we also know at which timing the flower development is aborted or promoted. So it's meiotic timing, pre-meiotic timing, so in stamen, when the differentiation begins. So the genes that work at that stage will be the first candidate for as the sex determining gene.
So the identification of these important genes was based off the RNA sequencing on that particular timing.
Yeah, but specifying that particular timing is dependent on the previous studies.
Previous study on histological analysis.
And I appreciate those studies.
Indeed. So thank you, Hisham. And in the remaining time, I would like to welcome questions from the Zoom participants. I believe we have some here. The first one should be from Andika, the online participants. It's below you. So I have experience analyzing Illumina data consisting of contigs to construct a reference genome, but there are unplaced contigs that cannot be inserted into chromosomes. Is using PacBio will be a solution for this kind of problem? That is his first question.
Yes, yes. PacBio will help
PacBio will help
very often.
with some high budget issue. Okay. And the second question would be, how do you analyze NGS data that doesn't have a reference genome at all? And if for RNA sequencing analysis, there is no reference gene annotation available for the RNA sequence analysis. So basically, if you don't have reference, how to analyze the NGS data?
It is
Or no reference genome, then how to analyze the NGS data, because you need to map the reads to reference, right?
we will do de novo assembly of reads into contigs.
De novo assembly.
Somehow we will try to get the longer contigs using the de novo assembly of reads, and then we will use it. The resulting sequences as a reference.
Okay. So basically, we will develop the reference by de novo assembly. So in the context development, you require particular or some overlap between reads, right?
Yes.
Is there any requirement? How long should be the overlapping sequences for the de novo assembly?
I don't remember how much overlap is necessary for the de novo assembly, but the data amount should be like 20 times at least for the genome. For genome assembly, 20 times more data than the reference genome size will be necessary. For example, 20, for example, for 1 gigabase genome, 20 gigabase data should be necessary.
Okay, that's for the de novo assembly. Okay, so I hope this answers answered Andika question. So do we have any other question from the list? From Rita, from your own opinion as an expert, does any genetic assessment nowadays always require NGS? Does any genetic studies always require NGS?
I don't think so. Classical marker can be powerful.
Especially because many publicly available data is
that's right. Yes, that's also true. And publicly available data are useful. And to me, NGS data are easier to handle than doing experiments. Personally, I have no time for doing experiments for genotyping, but I have some time for browsing NGS data and analyze them. So that may tell the situation probably.
So yes, the audiences here will also note that our molecular study in genetic analysis doesn't always require NGS directly, because there are some NGS result data that is publicly available that can we use. And we can also still use classical approach like
I think
markers.
Classical approach still can cost some, right? To order primers and to order enzymes, and a lot of time will be used. And for NGS, we extract DNA and send it to company. So it's handy in a way. And the budget is becoming lower, but required.
Yeah, compared to the output resulted, right?
Yeah.
The second question from Rita is, how can we know such analysis is still relatively costly here? Or maybe she's asking about, do you have any other methods that will be similar with NGS in the output, but more reliable in cost?
What does it mean?
Yeah, NGS with similar, do you have any suggestion for other methods other than NGS that can provide similar informations, but more affordable research price? Yeah, simply, I think that is.
I think NGS is more affordable than the other methods. Like, for example, for transcriptome, microarray is available, but I think microarray requires a lot of experience, and if we use company, then the cost will be higher than NGS. So microarray is not very good option for, at least for me. And for genotyping, some tiling arrays or snip array is available, but the same thing applies, it's array based. So, あとは yeah, I think NGS is better.
So until recently, then the NGS is the more affordable technology for such powerful output compared to the existing technology right now.
Just
The NGS, right?
NGS.
Yeah. Okay. We have any other questions from the Zoom? Interest to the online participant, she is interested in the math map analysis. You explained about the math map analysis.
Map.
Multiple something, right?
Okay.
The question is the mutant which is used as one of the parental crosses is M1 generation or have to be a stable mutant. Okay, you explained to cross the one species with the mutant. So what is the requirement of the mutant? It should be in the, can be it be in the first generation of mutant, or it should be stable in the later generation to be crossed?
I think it can be the first generation.
First generation is okay.
The point is that we should be able to obtain F1 plants from the cross, and the phenotype segregates in the F2 generation. And the benefit of this mute map analysis is that we can use the F2 generation for the analysis. So if the even if the phenotype is fatal or lethal, we can use those mutants. If they are somehow alive, even if it's very small, we can collect them as samples for sequencing. So that's a benefit, and that's true for M4 or 5 generation.
Can be also applied.
Yeah.
And when you do the sequencing of the F2 generation of this crosses, do you also need to sequence the parental?
I think it's necessary. I have ever seen the protocol, and I don't remember it well, but I think it is necessary for sequencing the parental lines as well.
And do we also need to do the phenotyping at the same time to observe the phenotype of these F2 segregants?
Yeah, we have to see the phenotype at least like 50 plants or so in F2 generation to, we have to get a bulk. I think one bulk consists of like 20 plants or so in that protocol, original protocol. So
20?
10 or not 10, like 20 or so. So relatively small number of plants are used as bulk.
For the mutant.
For the mutant.
But for QTL-seq, how many? More?
Not large.
Not large? Okay. I was questioning about that. If I have the bulk, how many plants in the contrasting phenotypes group should be bulk?
But I think it can affect the result. But I sometimes see some non-sharp signal in the QTL-seq or mute map SNP index, and it may be dependent on the sample quality. So.
Okay. Do we still have questions? Yeah, okay. It seems that we have many questions from the Zoom. Maybe another one. We still have remaining time from Yulita. She's asking about the dehuling trait on your soba, on your tartary buckwheat. Could you explain a little bit more on how a deletion in the mRNA causes 14 amino acid deletion and should derive from an SNP in a gene? Okay, I believe you show one SNP. And this one SNP causes 14 amino acid deletion. How could it be?
Okay, so first I will explain again the step. of this analysis. We
Can-
first
Can you please show the slide
okay,
of
that will help.
the gene diagram and the SNP position of the tartary buckwheat? A moment, please. So more to the tartary buckwheat. Last part, I would say. もうちょっと. Again. Next. Again, next. Yes, that one. The previous one. This one. Okay, thank you very much.
So what we identified first is the 42 base deletion. In the former slide, actually, one former slide is showing the 42 base deletion. It was not saying that it's 42 base, but the deletion detected in the difficult Herring cultivar was 42 base deletion. And because it is 42 deletion in the coding sequence, we assumed or we thought we didn't analyze the protein sequence of this from this gene in that cultivar, but because it's 42 base deletion, we thought that this is 14 base amino acid deletion. Okay, so this is just an prediction. No direct analysis was performed for the protein. So 14 amino acid deletion was something like that. Derived something by that kind of idea or the situation. And we cloned the sequence of the genomic sequence of this gene. And so full sequence was determined for each of the cultivar used for this analysis. And we compared those sequences, and this was the only difference, one base difference. And this model was developed by the difficult. Sorry, I think I was confused. And this is based on the model from the tartary buckwheat genome. And that sequence was consistent with the difficult Herring cultivar, Hokkai, this cultivar.
Yes.
With this amino acids. And so this is
Easy
this is
rehaling.
corresponding to this one, the bottom one. And this SNP or mutation was located in the here in the very end of the intron. It's a splice acceptor site.
Yes.
This was component of the splice acceptor site. So, and this was 42 base, this exon. Can she or he?
The operator helped to point it.
Thank you. And so this exon was 42 base. So we think that this didn't work as the splice acceptor site in this cultivar, and then this was skipped.
Yes.
The splicing was like this, skipped, and then that caused the 42 base deletion messenger RNA, and that caused 14 amino acids in protein.
So I would say I would like to confirm my understanding. So the SNP in the exon intron junction caused the alternative splicing. In this case is exon skipping, and the exon land is 42 nucleotide, so it removed the 14 amino acid, right? Okay.
Yeah, and I think some other examples of this kind of situation are available in papers. So
Fukui et al. 2020, if you would like to read further.
Some mutant analysis using rice or some other publications are, I have no specific example, but
Indeed.
The mutation in the exon-intron junction can cause this kind of situation.
Thank you very much, Tsugama Sensei. I believe we still have some questions, but unfortunately we come to 11 a.m. by now. Time is very limiting, but Tsugama Sensei will stay in our department until Friday. So if any of you have the opportunity to discuss further with him, we are welcome with that. Okay, we are happy. So with that saying, I would like to thanks again, Tsugama Sensei and all the audiences for your fruitful discussion and active discussion. And I'm returning this session to our Master of Ceremony today, Mas Ade. The time is returning to you. Thank you very much.
Terima kasih very much.
Thank you, our speaker, Dr. Tsugama, and our moderator, Dr. Sinto, for this productive session. Before we move on, it's time for us to extend our great appreciation to our speaker and moderator. And we would like to request our speaker and moderator, okay, to please stand up. Okay. Yes. To present the token of appreciation, we would like to invite the Department Head of Agronomy and Horticulture, Professor Eddie, and accompanied by the Head of Study Program of Magister Program. Yeah, Bu, mangga. Yes, Professor Dewi. Yes, please to come forward. And for the committee, take your position for this documentation session. Okay, first appreciation will be given to our speaker, Dr. Daisuke Tsugama. Okay, thank you, Dr. Tsugama, for this presentation. Okay, this is the certificate and also the token of appreciation that given by Professor Eddie to Dr. Tsugama. Thank you so much. Okay. Next, okay, to the next appreciation. So the next appreciation will be given to our moderator, Dr. Sinto. Thank you for guiding us in this amazing session, Dr. Sinto. So excellencies, please gather in the middle because we will capture this moment. Okay. Okay, let's give your best smile. Okay, already it's done. Okay. Okay. Thank you very much, excellencies. You may return to your seat. And let's give them a warm applause again. Okay, thank you very much. Okay, excellencies, eminent speaker, ladies and gentlemen, our guest lecture is completely over. Thank you very much to speakers and moderator. And for our on-site participant, we kindly remind you to fill in the form of the attendance through this link and QR code provided. And for our Zoom participant, the link has already been shared in the chat box. You may fill your email in the form because we will give you the certificate for free. And last but not least, on behalf of the Department of Agronomy and Horticulture, Faculty of Agriculture, IPB University, we would like to extend our gratitude to everyone for this awesome cooperation during this guest lecture. And then I would like to ask all participants in the Zoom meeting to open your camera right now because we will have the documentation session after closing. We wish a pleasant day. I'm Ade Buhabi signing off today. See you and wassalamualaikum warahmatullahi wabarakatuh. For the participant on site, I would like to invite you to the front to take a position to the documentation session. Also for our all lectures, Dr. Willy, Mr. Eric, Dr. Zulfikar, and Professor Dewi Sukma, Professor Eddie, Dr. Ning, all the lectures and all the participants as well. Please gather in the middle. All the participants also. Don't be shy. Okay. 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 For the attendance form, we will try to fix it. Please wait and try to open the attendance link again. And For all the participants in the Zoom meeting, open your camera because we will take for the documentation session as well. Okay. Because we will, while we wait for the participant on site to take their position, first we will take the documentation from the Zoom meeting. Okay, are you ready from the Zoom meeting participant? Please open your camera. Okay. Oh, boleh Bu, dosen dulu aja. Okay, first maybe for the lecturers and the speakers and also the moderator for the committee, please take your position there. Okay. Yeah. Maybe Pak Eric, maybe take your position there. Okay. Bisa, sana ya. Okay, let's give your best smile. Ready? 1, 2, 3. Okay. Another pose. Okay. Okay, for all the participants, we kindly invite you to the front. Take your position. Maybe for who have like not really tall, maybe we can sit in the front. Okay. For our online Zoom meeting participants, sorry. Okay, we will take for their documentation as well. Okay. Please open your camera again. Okay, we waited until 5 seconds. 1, 2, 3, 4, 5. Okay, take your position and give your best smile. Okay, thank you. How many slides? 3. Okay, first we will take from this first slide. Okay, so 1, 2, 3. Okay, next slide. Okay, there's no open the camera, but still okay. Okay, 1, 2, 3. Okay, because we already fixed the attendance form, so you can access the attendance by the chat box. And please fill the email in that form. Okay, for our on-site participant, okay, you already take your position and give your best smile. Maybe we can turn on the light first because some of our lectures cover the light, their face cover the light. Oh no, I mean the projector like. Oh. Okay, thank you, Dr. Sinto. Okay, ladies and gentlemen, please take your position and give your best smile. 1, 2, 3. Okay, with another pose. Freestyle. 1, 2, 3. Okay, awesome. Okay, thank you very much, everyone. Okay, maybe because the projector is not working, I think, oh, already. So for our on-site participant, you can access the link, the attendance link through this QR code to fill your email there to help the committee to send you the e-certificate for free. We would like to remind you again to fill the attendance form through the link provided and QR code provided there in front of. So you can scan the QR code to access the form to fill your email so the committee can send you the e-certificate. And for the certificate will be for free.
Juni, tapi ini kita masih mencari peserta. Jadi pesertanya 17. Iya, jadi ke IOP itu kan kita mendaftarkan 20 abstrak untuk IOP-nya. Terus kemudian dengan IPB-nya kontraknya 37 abstrak yang berafiliasi ke 80. Iya, kegiatannya sama seperti